Yeah, I'm a pediatric oncologist and I take care of kids with cancer at Alberta Children's Hospital. The problem need that we are trying to solve is a treatment of an unmet, or coming up with a treatment for an unmet need in pediatric oncology as well as adult oncology. Overall, that we do well. Most of our children get cured and go home, but there is a small number of cancers that we do not cure at all, especially the ones that affect little babies or newborns. So this has been an interest of mine for a very, very long time. About 10 or 15 years ago, I had a little baby die in the hands of the mother, and I made a promise that we will work on that cancer. So it had been a long road, only because we were looking at the conventional things, a mutation or an oncogenic driver. But what we found in the recent years is actually a protein that shows up that should not be there. It should have disappeared when the baby formed, but it persists. What it does, it blocks pathways that overcomes the tumor formation. In other words, it takes away a tumor suppressor gene function. So what we did, then we went on to design, using molecular modeling and other technologies, a drug that would bind to this protein and essentially take it out of circulation and let all those tumor suppressors come into play, and the tumor disappears. So after spending many different types of the molecules and optimizing their function, their toxicity, and ability to cross the blood-brain barrier and so on, we have arrived at a molecule. And so this is one example that, so we created this tumor in animals as well as in the lab, and give doses of this medication. As you can see, it essentially takes away the formation and survival of the tumor. So we went on to file a patent. We received a patent on this molecule recently, a full patent. So what we have found, interestingly, is this pathway is not unique for this with very rare pediatric cancer. It's also found in a number of other adult cancers. All of them are currently incurable. Breast cancer, prostate, colon, and so on. A small percentage in each cancer, but it's still there's an unmet need to treat these patients. So our evaluation of the market showed about 45,000 women will die from breast cancer every year, and 2,180 may have benefited from a similar drug. In other words, this pathway occurs in about 2,000 or 3,000 of these patients. A total of about 20,000 of all cancer-related deaths in North America can be prevented by this drug right now. And most importantly, there are no other drugs that we know of or in development to target this pathway that's clinically usable. The talent readiness, we have education, training in biochemistry, molecular biology, experience over 30 plus years of working in cancer field. And we have also worked with commercializations, especially the drugs developed by other companies. And the skills that we have are project management, supervision, team building networks within the university, and information management. So why us? Because we have direct clinical experience, we are able to translate the information about the drug to clinical trials. I do clinical trials as a major part of my work for kids with cancer. So we have the network and the ability to write clinical trials and do clinical trials in the future. And we have significant community support, anything from the Kids Cancer Care Foundation in Calgary or the foundation in Edmonton, as well as parental and institutional funding across the globe. So our company structure is co-founder and researcher, and we have scientists who are working, and we also have managers working with us with respect to finance and managing the project. We have a number of people in our group that we have received awards from the Innovate Calgary. Today I got the Tenant I2C program award. that brought in the expertise of a number of people. So Innovate Calgary, Genome Alberta, and as well as CEOs and developmental scientists from large pharmaceutical companies like Provectus are part of our team. So this is our business plan. So we have completed the early pharmacology, clearly described the on and off target effects. We have clearly described the mechanism of action. And preliminary safety studies in animals, small animals, and large animals are in progress. And we have filed a PCT on the structure, synthesis, activity, and the various uses of this drug. So right now we are looking to synthesize this agent in a GMP facility. When I was talking to my adult oncology colleagues, there is a type of adult cancer when it not respond to the current chemotherapeutic agent, almost all the patients go into palliative care. So there is a need and patients available to do a small clinical trial. So we are in the process of writing a clinical trial to use our drug after being synthesized in GMP to generate the very first set of safety and clinical data. Ultimately, what I would like to do is to take this on and work with large pharmaceutical companies for acquisition and licensing of this agent to bring it to the clinical trial. So in effect, what we want is to have this drug into clinical trials and into licensing as fast as we can, because right now we are not curing these patients at all. So what we are asking is investment for the GMP production and subsequently to do the clinical trial at Tom Baker Cancer Center and Edmonton to start with, because we do have the patients available and the adult oncologists are willing to start a clinical trial with this agent. Introduction, connection to pharmaceutical companies or investors to bring this to the next stage to take it to commercialization. Thank you so much, and I'll answer if you have any questions.
All right, thank you so much, Aru. Feel free to unmute yourself if you have any questions or ask them in the chat space. I have one question that I noticed on your previous slide. Is you noted GMP production was important. Could you discuss like what that is and why it's so important to your process?
Yes. So right now we make this drug in a company in Edmonton that's not licensed by Health Canada or FDA to be used in humans. So to synthesize the same molecule, it'll cost more money. And there are companies here in Canada as well as in the US will take it and synthesize the chemical for us, the drug for us to use in humans. So that is the next critical step, because that's required by Health Canada for human use.
Great. Can I ask a question as well?
Sure.
Very impressed about the story. I'm just wondering about your target selectivity. How are you certain you're on target and not hitting significant off targets?
Yeah, so that's a very good question. So we have generated a number of cell lines that carry these targets from our patients as well as through collaboration from others. We also went ahead and silenced this protein using siRNA technology or transfecting this protein into normal fibroblast cells and show they become malignant. Then we treated them with the drug to show that the effect goes away. And then we have other tumors exactly the same, say breast cancer, without this pathway, and we showed that our drug had no effect on those cancer cells.
Great. And the mouse studies, I guess you've tried different xenografts, is that right?
Yes, we transplanted almost all these cell lines, 5 adult tumor cells and pediatric cells and cell lines, and show, providing you have the pathway there, it'll go away. And also, it's not that difficult to test the presence of this protein. A very simple Western blot can identify that you have your target in the tumor.
Okay. And have pharma shown any interest, or are they just waiting till you get more data?
I have not approached any pharmaceutical companies yet, other than in my regular work. I work with Roche. We are running a few clinical trials for different companies. When we talk about it, they showed interest. Bayer showed interest. They wanted me to sign an MTA and talk about this, but I kind of pulled back because I need more advice as to how to proceed. And also, we just filed the full patent. Before that, we had only the provisional patent. So now I'm a little bit more comfortable taking to discuss with pharma. But the advice I was given by different people in our committee is that you need to have this small but solid clinical data for pharma to get interested, because they probably get 250 drugs coming to their pipeline every year. So they want, if you want them to be serious, then you have to show clinical trial data. So I'll be happy to take any advice you guys can give us with respect to that pathway.
Sure. There's some great questions by Christy and also then John and Svetlana in the chat space. Do you want to respond to those?
I stopped.
The first is, have you spoken to API, Health Cities, University Hospital Foundation, Edmonton, I suppose also in Calgary? What's been the response there so far in those organizations?
I have not talked to them, but I'd love to get those names and contacts for sure.
Okay, well, maybe Christy can follow up with you on that. And then John also asked a question. John, do you want to ask?
Sure. Can you hear me?
Yep.
Hi, John. Yes.
Yeah, sorry. I was just looking at your proposed phase 1 with an N of 10 patients. I'm wondering if you couldn't consider adding either other indications or other preclinical evidence to expand that phase 1?
Yes.
Because, you know, if this can be used in other cancer, adult cancer, breast cancer, as you suggested, then why would you not look at that?
Absolutely. I mean, the cost is a concern. And also what we are trying to do is the first set of studies, the phase 1 clinical trial is mostly toxicity data. So 10 to 20 patients, I kind of revised that 10 to 20 with our last meeting with the experts. But they suggested that should be enough to complete an effective phase 1 clinical trial. Ethically, I don't know if I would include placebo, if that's what you are getting at, in a group of patients without this target. That would be a perfect scientific experiment, but may not be very ethical, because we know for sure this drug absolutely has no effect unless you have that target protein in those tumors.
Yeah, so I'm just curious that, you know, what, in that case, then what would your primary indication be moving forward?
The toxicity, tolerability, and of course, if we get any data on tumor regression, we will include that in the data, but the objective would be to determine the dose-limiting toxicity and an effective dose to go forward for the phase 2 of the clinical trial, or take it to the pharmaceutical company. So they can either have me as a partner, because we do design drugs and trials. We have patients. I'm part of a consortium that includes MD Anderson and Harvard and so on. So we have the patient population, even for rare tumors. And these patients, these kids absolutely have nothing to go for. So the parents will be very willing to enroll in a clinical trial, providing all the safety and other concerns are addressed and watched very carefully.
All right.